This strain is responding well to the new medium. Good growth on the controls. Confirmed, I'll log the results. Noted. The streaks are showing good separation here. Now to check for contamination. The colonies on this plate look healthy. We'll isolate this one for further study. I'm collecting a sample from this colony. Now I'll streak it onto this fresh plate. This prevents contamination and isolates the bacteria. Now we can incubate it. Observe the colony morphology here. Now compare with this sample. Note the differences in growth. Observe the streaking technique. We use a continuous motion for isolation. This ensures single colonies form for analysis. Understood. The angle is crucial. Exactly. Now we incubate. That streak looks very clean. Yes, a perfect technique. Thanks. Now for the next sector. What's the growth projection? We should see colonies by morning.